Good afternoon everyone, I'm Chua. Today we are going to talk about Mendelian genetics. So these are the learning outcomes of the lecture. Students should be able to explain the Mendel models. Besides that, students should be able to use the monohybrid and dihybrid cross to explain and study the pattern of inheritance. And lastly, students should be able to explain all these genetic terms. So this is a preview of the lecture. So first we will talk about the Mendel's model, which include the monohybrid cross and the dihybrid cross. Monohybrid cross is when we track one character, while dihybrid cross is when we track two characters at the same time. Then next we'll move on to the Mendel's law. So there are two Mendel's law. The first law is the law of segregation, and the second law is the law of independent assortment. We will also talk about the lean gene. Lean gene is a gene that don't follow the independent assortment. These days, scientists are able to calculate the probability of you to get an inherited trait or genetic disease based on the information that they have from your parents and family history. So the genetic disease include albinism, the Huntington disease, sickle cell anemia, and the color blindness. So how was this possible? Well, we have to thank an Austrian monk who loved the plant very much. Well, he is the one that made all of this thing possible. And his name is Gregor Mendel. Mendel is the first person to analyze the pattern of inheritance. He managed to discover the basic principle of the heredity by just breeding the garden peas without the help from any advanced equipment like the computer or the calculator. What he had is just his mind. But Mendel studied garden peas. Well, garden peas is good because they are easy to grow, they grow very fast. And they also have a lot of variety that allow us to observe. And most importantly, they are easily manipulated. We can simply transfer the pollen from the stamen of one flower to the carpel of another flower in order for fertilization to occur. And this garden peas can self-fertilize as well. Mendel tracked 7 visible features, including the color and the position of the flower, the color and the shape of the seed, the shape and the color of the pod, and the stem length. And this is the result that Mendel found from his experiment. If you are Mr. Mendel, what are the conclusions that you can make from this result? Well, Mr. Mendel made 2 conclusions. The first thing is, he found out that when you crossbred 2 individuals with different traits, and you will produce the offspring with only one trait, one of the traits will be disappeared. And the second thing is that when you crossbred this offspring, the trait, that the so-called missing trait, will reappear again. And the ratio is always 3:1. Mendel actually did the same thing for every single character, and he found the same result. So Mendel started with the monohybrid cross, which means he only looked at one character. So in this case, we will use the color of the flower as an example. So when for P generation it means parental generation. It is the first generation in the Mendel experiment, in the Mendel cross. And F1 generation is the first filial generation. So this is the result when you crossbred the P generation. And when we crossbred the F1 generation, we will produce the F2 generation. So in P generation, Mendel crossbred one purple flower with white flower, and the offspring that produced is all purple flower. And when he crossbred the two individuals from the F1 generation, he will find out that 3 out of 4 of the plant is having the purple flower, and only 1 out of 4 of the plant is having the white flower. Now during Mendel's time, everybody actually believed in the idea of branding. So the idea of branding actually states that when you crossbred two individuals with different traits, the trait will be branded and passed on to the next generation. So there will be, it is weird to see a white flower, right, in the F2 generation, because both of the parent is purple color. So they shouldn't pass on any white color trait to the offspring. So this will also means that the idea of blending is actually wrong. But in P generation, the purple flower will have 2 big P, and the white flower will have 2 small p. So what are the gametes that these two individuals is able to produce? Well, for purple flower, they are only able to produce the gamete with big P, while for white flower, they are only able to produce the gamete with small p. So when you fertilize, when fertilization occurs, they produce an offspring that contain 1 big P from this parent and 1 small p from this parent. So all of the offspring will contain 1 big P and 1 small p. So what are the gametes that this individual is able to produce? Well, they have 50% of chance to produce the gamete with big P and another 50% of chance to produce the gamete with small p. So in other words, the individual in F1 generation are able to produce 2 types of gamete. So these are the results when fertilization occurs. So when the gamete with big P fertilize with the gamete with big P, then they will contain 2 big P, and this is the so-called homozygous dominant, because they are containing 2 dominant alleles. And when the gamete with big P fertilize with the gamete with small p, then they will be the heterozygous. This means that they have 1 dominant allele and 1 recessive allele. And when the gamete with small p met with the gamete with small p, then they will be the homozygous recessive, because they are containing 2 recessive alleles. But you can notice here is that the phenotypic ratio and genotypic ratio will be different. The phenotype is what we can see, what we observe. And genotypic ratio is, genotype is the genetic makeup of the individual, which we can observe with our bare eye. So for phenotypic ratio, you will see there will be 3 to 1, 3 purple and 1 white. Well, for genotypic ratio, it will be 1 to 2 to 1, which is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Well, we can link all these things with the meiosis and the fertilization, because meiosis is actually the process of gamete formation. Well, this is the 2 individuals from the F1 generation. So F1 generation, they are both heterozygous. That means that they are containing 1 small p and 1 big P. So same thing goes to this individual. So the gamete that they can produce during the meiosis is, they will produce either gamete with small p and gamete with big P. So they have same thing goes to this individual. They have 50% of chance to produce the gamete with small p, 50% of chance to produce the gamete with big P. So fertilization will occur randomly. So that means that the gamete with small p might fertilize with the gamete with small p. It can also fertilize with the gamete with big P. Same thing goes to this big P. it can fertilize with this, or it can be fertilized with this as well. So the outcome, if this small p fertilized with the small p, it will be the homozygous recessive. And if this small p met with this big P, then it will form heterozygous. And if this big P fertilized with this small p, it will be the heterozygous as well. And if this big P fertilized with this big P, then it will form the homozygous dominant. So when you look at it, the phenotypic ratio is still 3:1. 3 purple and 1 white. And the genotypic ratio is 1 homozygous dominant to 2 heterozygous and to 1 homozygous recessive. So from the monohybrid cross, Mendel actually came up with the first law, the law of segregation. So Mendel stated that the 2 alleles for each gene will separate during the gamete formation. In other words, every individual will receive 1 allele from each parent. So Mendel also carried out dihybrid cross. That means that Mendel will follow 2 characters at the same time. So in dihybrid cross, Mendel came up with 2 hypotheses. The first hypothesis is hypothesis of dependent assortment, and another one is the hypothesis of independent assortment. In hypothesis of dependent assortment, it means that the gene that control the 2 characters will go into the same gamete in the package. Well, for hypothesis of independent assortment, it means that the gene that control different characters will go randomly. So this is the 2 hypotheses that Mendel came up. So in hypothesis of dependent assortment, that means that the heterozygous individual in the F1 generation, they can produce only 2 types of gamete. Why? Well, these are the type of allele that present in the F1 generation. So if it is hypothesis of dependent assortment, that means that the dominant Y and dominant R will always pair up. And the recessive Y and recessive R will always pair up. So that means that there is only 2 types of the gamete that can be produced. But if under the hypothesis of independent assortment, the big Y can pair with big R, big Y can also pair with small r. Small y can pair with small r, small y can also pair with big R. So that means that the individual is able to produce 4 types of gametes. So if according to the hypothesis of dependent assortment, that will mean that the phenotypic ratio will be 3:1, 3 yellow round and 1 green wrinkle. Well, for hypothesis of independent assortment, the phenotypic ratio will be 9:3 to 3:1, 9 yellow round, 3 green round, 3 yellow wrinkle, and only 1 green wrinkle. And guess what Mendel found? Well, this is what Mendel observed in his experiment. He found 315 yellow round, 108 green round, and 101 yellow wrinkle, and only 32 green wrinkle. And the phenotypic ratio is approximately 9:3 to 3:1. And this means that hypothesis of dependent assortment is wrong. So with the result of dihybrid cross, Mendel actually developed the second law, which is the law of independent assortment. This second law states that each pair of the allele will aggregate independently of the other pairs of allele. So this also means that the inheritance of one character will be having no effect on the others. So that's why you can see those people with hitchhiker thumb will not necessarily have the free earlobe. Those with the free earlobe will not necessarily have the middle peak. Why? Because all of these genes that control this character will be segregated randomly during the formation of gamete. But wait, I think some of the people will notice that those people with red hair, naturally of course, not artificially, those people with red hair, they will have the freckles at the same time, most of them, right? So, strictly speaking, the second law, the law of independent assortment, only apply to those genes that occurs on different chromosomes, on non-homologous chromosome. And if they occurs on same chromosome, then this gene should be far apart. And if, let's say, the gene occurs on the same chromosome and they are very close to each other, then all of this gene is called the link gene. So what are the link genes? Well, everyone knows that we inherit half of the chromosome from our father, half of the chromosome from our mother. So after fertilization, so this is all the chromosome that we have. So you look at the chromosome, the gene D and gene E. So they occurs on non-homologous chromosome. So during the anaphase 1, when it is being separated, they will be separated randomly. So they will follow the law of independent assortment. Then for gene C and gene F, they occurs on the same chromosome, but then they are far apart. So same thing, they will follow the law of independent assortment. But for gene A, B, C, they are on the same chromosome and they are so close to each other. So during the anaphase 1, when they are being separated, all of these genes will tend to go into the same gamete. So this will mean they are all the link gene. The A, B, C are the link gene. And this might explain why those people who have the red hair will have the freckles at the same time. Because the gene that control for freckles and the red hair, they occurs too close to each other on the same chromosome. So this is a definition for all the genetic terms that I think will help you a lot to understand all these terms. And with that, that's all about the lecture today, and thank you.